Good afternoon, everyone. It is my pleasure to introduce Dr. Ayotunde Dokun as our speaker today. So those of you online as well, hopefully you can hear me well.
The things that interest you, that you have a passion for, and it'll keep you asking interesting questions. And so while my PhD was in immunology, the key thing I took away from that was learn how to be a good scientist so that you can apply to whatever your interests might take you in future. So for the trainees, postdocs, or graduate students out there, that's my word of advice is learn how to be a good scientist, because the things that you're studying today may not be what you spend your career studying in future. The tools that you acquire during that training becomes what you have to keep applying. So I'm going to be talking about exploration of mechanisms contributing to poor disease outcomes in diabetic peripheral arterial disease. Let's see if I have this correctly. So to just tell you a little bit first about PAD, for those who may not be familiar with PAD, it's the presence of occlusions in the blood vessels other than the heart. This occurs most frequently in the arteries of the lower extremities. There are two classic presentations of PAD, but before I go into that, let me just quickly show you this, run through this angiogram of a patient who has PAD. You will notice that there's no flow here and no flow here. This is a patient with complete occlusion of the femoral artery, and this individual's limb is still intact, and presumably because he was able to develop collaterals here, develop angiogenesis here, and is able to fill this limb and keep it perfused. I don't even know the patient knew that he had PAD because he adapted quite well in spite of having this massive vessel occlusion. So the classic presentations of PAD include intermittent claudication, which is what this gentleman is showing here, where patients have pain with ambulation, which unfortunately actually tends to make the condition, it's a chronic issue. And if you find out that you walk 2 blocks and have pain in your legs, often patients just stop walking 2 blocks. They just walk less. Um, and this is associated with rare amputation. 1 to 2% of these patients really require amputation. And also about 1 to 2% of these, the mortality is low in this patient population. The other classic presentation is called critical limb ischemia, or you may have heard limb-threatening ischemia. Critical limb-threatening ischemia is referring to the same condition. These patients tend to have pain at rest, and they may have ulcers, as in this guy's limb here, or they may have limbs that are gangrene, that has gangrene. So this affects millions of people in the US, and the numbers are even much larger worldwide. This is associated with high amputation rate, as high as 45% in some cohorts. What I find very interesting about this, though, is that the annual mortality for this patients who present with this is as high as 20%. To put that in perspective, many of the cancers that we hear and we're really scared about, don't have an annual mortality of 20%. And of course, we pump a lot of dollars into research and I'm not saying we shouldn't, but I think that there are things like this that we ought to consider pumping some decent amount of funds into supporting. And this is actually, some of you who are endocrinologists may have already seen this in your practice. I was in service just last week, and part of one of the patients that I was consulted to see for glycemic control came in with critical limb ischemia. And of course, they did the angiograph and tried to intervene or observe the occluded vessel, but nothing changed. They opened the blood vessel because it's most likely a microvascular disease. So perfusion didn't change because they opened up a large vessel. What we couldn't tell him during that visit is that he's most likely going to lose that limb. After a few days of trying a variety of things, there's not a whole lot you can offer him other than open up the big vessel. If the limb continues to be ischemic, eventually it becomes gangrenous and they will amputate it. So the risk factors for PAD are very similar to that of CAD, except that PAD, of course, is less studied. However, there's some distinct differences in terms of the risk factors. In PAD, diabetes and smoking actually accounts for 80% of the risk of developing the disease, which is not the case in the case of heart disease. And when you look at the effects of diabetes on PAD severity, PAD patients with diabetes are 5 times more likely to develop critical limb ischemia. And when you look at the risk factors for developing critical limb ischemia, diabetes has the biggest impact. So of course, this is one of the reasons why this endocrinologist who's interested in diabetes is interested in trying to better understand this. When you think about the role of diabetes in PAD outcomes, there are a number of ways to think about this. You can think about the metabolic derangements that are associated with diabetes leading to accelerated atherosclerosis. These same mechanisms contribute to impaired endothelial function, but they can also impact adaptation following vessel occlusion. I showed you the angiogram on my very first slide of a patient who had complete vessel occlusion, did not even know that he did, because somehow his body adapted to this occlusion, created collaterals, got blood flowing to the tissue, and he's walking around. So this impairment of adaptation following vessel occlusion is an area that's been of interest to my group. Like many forms of human diseases, PAD, there's been a mouse model of it that's been developed, and that's what I'm trying to show here. Essentially, what you do in the mouse model is you ligate the femoral artery proximal and distal to the popliteal bifurcation, and you excise this segment. Right? Following that, the tissue distal to that excision of the femoral artery will then develop ischemic injury, which, depending on the circumstances, the mouse will either recover or lose the limb. And we can then study both blood flow recovery in this tissue that distal. We can look at gene expression, microRNA expression, protein expression. We can look at a variety of things. People who are interested in looking at collaterals or studying collaterals can look at upstream here to look at collateral formation and the processes involved in that. So when you think about the strategies to then employ to try to understand how diabetes might impact PAD outcomes, we've used a number of different approaches. The first one I'm going to talk about is looking at established pathways. So what do I mean by that? So there are certain factors such as VEGF or vascular endothelial growth factor that's been studied extensively as a pro-angiogenic agent. And so we've looked at, so what happens to this pathway or this signaling pathway in the context of a diabetic PAD. And while a lot of prior studies have actually looked at VEGF expression, the protein expression, and mRNA expression in the context of diabetes, our studies was one of the first to look at the VEGF receptor itself. And what I'm showing you here is a summary of essentially the paper that we published related, one of the papers that we published related to this, where we show that increased ubiquitination of VEGFR2, both in vivo and in vitro, impaired VEGF function. So what you see here is a Western for VEGFR2 expression, and we've immunoprecipitated that receptor and looked for the extent of ubiquitination in mouse with diabetes, and you can see that there's increased ubiquitination. That's what's quantified here. We've also shown that if you actually treat the diabetes, so mice that we induce diabetes in, right after induction, we start giving them insulin, you can actually reduce the amount of VEGF receptor ubiquitination. This is probably the more important slide here. In vitro, looking at endothelial cells, you see that there's decreased VEGF receptor expression. But if you use an inhibitor of ubiquitination in a dose-dependent manner, you can actually see restoration or preservation of the VEGF receptor. That's probably better shown here, where hyperglycemia or high glucose treatment of the endothelial cell results in loss of VEGF receptor 2 expression. Inhibition of ubiquitination restores that expression. Other pathways that other approaches that we've taken to try to understand this better include looking at novel pathways that have not necessarily been shown to be critical for angiogenesis or post-ischemic adaptation. To do that, we've exploited genetic differences in adaptation to ischemia. So to that effect, this is one of the studies that we've done that's summarized here, where we've exploited genetic differences in adaptation to ischemia. So the black 6 mouse, if you induce ischemia using, or experimental PAD using the femoral artery ligation that I showed you, they have a tendency to recover very well. These mice over 2 to 3 weeks will start using their limb as if you never operated on them. However, the the biopsy mouse has poor perfusion recovery, as you can see here, and they have a tendency to develop limb necrosis. And often that limb would auto-amputate over time, reminiscent of what you see in some humans that show up with critical limb ischemia versus people who happen to adapt quite well on their own to vessel occlusion. So using these two strains, we actually we did a classic genetics, crossed the two generator F1s, back crossed those two, the B6, the Bob C strain, generator N2s, did a classic genetic mapping and identified a genetic locus or QTL that we termed the limb salvage QTL1, located on Y chromosome 7. Within that locus, there are several genes then that we identified as potentially contributing to this, the ability of the block 6 mouse to adapt well to the ischemic injury. One of those genes is ADAM12, or a disintegrin and metalloprotease gene 12. Its expression, we later show, plays a critical role in the preservation of the limb in these mice, and this appears to occur through its effect on improving angiogenesis. We also have data that show that a SNP within ADAM12 is associated with critical limb ischemia in humans with PAD. I'm going to come back to ADAM12 later in the talk. Interestingly, however, the block 6 strain that I showed you that recovers very well and adapts quite well, if we induce type 1 diabetes in this, if we induce diabetes in these mice, whether using the type 1 model where you knock off the beta cells, whether with STZ or genetically in the Akita model, or if you place these mice on half-fat diet so that they become obese, you can do this plus or minus STZ to make it more of a type 2 model. When you induce high limb ischemia, this is what you get. They're more likely to lose their limb. In that model, when we look at the ischemic muscle here and look at ADAM12 expression, you can see that unlike the non-diabetic animal that gets a robust upregulation of ADAM12, ADAM12 expression is impaired. I'm going to come back to that. Another approach that we've taken is gene expression profiling. With our gene expression profiling, essentially what we're asking is, what are the things globally that's changing in ischemic tissue in a non-diabetic setting, with the assumption that those changes are probably part of why the mouse that's able to adapt quite well is adapting quite well. And then ask the question, well, in the setting where we don't see good adaptation, where the mice, in the setting of diabetes, where these mice are more likely to lose their limb, what happens to these same genes? And with that kind of experiment done, a number of genes were differentially expressed between the ischemic diabetic and the ischemic non-diabetic mice. Some genes went up in expression, some genes go down in expression. But when we ask the question, what pathways are affected, some were predictable. So metabolic pathways were affected. Genes involved in metabolic pathways were affected. But some were surprising, at least to me. Phagosomes and lysosomes, and then regulation of actin cytoskeleton. These are not pathways that I ordinarily would have predicted will be associated with impaired post-ischemic adaptation, which sort of gives you a sense of the value of this sort of forest view of the system of changes in gene expression.
Is this in the endothelium, or is this?
This is actually the whole muscle.
So, which makes a huge difference in terms of interpretation.
That is correct. That is correct. And the last approach that we've taken is microRNA expression profiling. And for those who may not be familiar with microRNAs, I thought I'd throw one slide in there that would give you a shot, some sense of that. So microRNAs are short non-coding RNAs, about 20 to 23 nucleotides that regulate gene expression post-transcriptionally, mainly by binding to a specific sequence of the 3' end of the mRNA, of the 3' UTR of the target genes. And this is just a schematic showing you that those microRNAs are transcribed, and then process as premieres and leave the nucleus, then part of what's called, eventually when they become mature microRNAs, become within the risk complex, in which AGO2 is a major protein there, which is where they actually interact with the mRNA that they're targeting. So looking at the mRNA profiling, this is basically a volcano plot that's showing different microRNA expression when you compare ischemic to non-ischemic tissue within the same animal, well, group of animals, it's not just one. What you see is that there are microRNAs whose expression go down, there are microRNAs whose expression are higher in the ischemic versus non-ischemic tissue. This particular microRNA has been flagged here, and I'll tell you why in a minute. When you do the same mRNA profiling, looking at mice that, same strain, but ischemic tissue from a mouse that has been put on a high-fat diet, you see that microRNA that was, whose expression was lower in ischemic tissue is now higher compared to the non-diabetic animal. And we've picked this particular microRNA because, those who do this kind of work know that you get a lot of microRNAs. How do you determine which one is of interest? Where do you go from there? You get a lot of data. We picked this particular one because it turns out miR29 actually targets ADAM12, which I showed you earlier we had worked on before, and it's shown that ADAM12 plays a role in this mouse's ability to adapt to ischemic injury. So to better understand how miR29 is expressed and to confirm our findings from the microarray profile, we looked, and you see in B6 ischemic limbs, you see less miR29 expression, and in the endothelial cells in vitro, you see less miR29 expression. If you then force expression of miR29, you see impaired ADAM12 upregulation. This is just the controls where ADAM12 normally gets upregulated robustly, as you see here, in the setting of decreased miR29 expression in the ischemic tissue or in the ischemic endothelial cell. Okay. The next question we asked then was, is miR29 regulating ADAM12 through direct interaction with ADAM12 mRNA, or is this something that's indirect? To get to that answer, we did a series of set of experiments in which we titrated different doses of an inhibitor to miR29 into lysates from endothelial cells, and then did AGO2IP. So basically, we pull down a protein complex that has both the microRNA and the mRNA interacting together. So essentially, as you titrate different doses of the inhibitor, you can pop off the microRNA as well as the mRNA from that complex if there's direct interaction. But if not, you wouldn't expect a dose-dependent loss of the microRNA or the mRNA. And that's what we found here. When you put in an inhibitor to miR29, you see a dose-dependent loss of the microRNA. Similarly, you see a dose-dependent loss of the mRNA for ADAM12. If you look at a control mRNA that's not targeted by miR29, you don't see a dose-dependent loss. If you use an inhibitor to another microRNA that's not miR29, you don't see a dose-dependent loss of the miR29 or ADAM12, suggesting that the regulation of ADAM12 by miR29 is through direct interaction. So we asked the question, I've shown you data that in the ischemic tissue or endothelial cells, you actually see less miR29 expression. So the question was, why is that important? Yes, we've seen that when we see under conditions where miR29 expression is low, you see upregulation of ADAM12. But physiologically, what's the implication in a mouse? Why does miR29 need to be downregulated? So we forced expression of miR29 and looked at what happened. So this is basically showing that we were able to achieve increased expression of miR29. When you look at ADAM12 expression, we're able to knock that down with that. That's the mRNA and that's the protein. Under these conditions, we look at with overexpression of miR29 in the ischemic tissue, you see impaired perfusion recovery, as shown here, and you see increased skeletal muscle myopathy in the ischemic muscle of the mouse. Then going now to, in the context of diabetes, what happens to miR29 expression? Well, in the context of diabetes, first we looked at ADAM12 expression. We can see that, as I pointed out before, ADAM12 expression is actually impaired in the ischemic diabetic model. This is a type 1 model. That's the ADAM12 mRNA. ADAM12 protein is also, expression is also impaired. Under those same circumstances, you see high levels of miR29 expression, both in vivo and in vitro. In vitro, in the context of high glucose, you see similarly high levels of miR29 expression in the ischemic endothelial cells. Is that a delay? All right. Then, because ultimately we're interested in relating this to human disease, we're fortunate enough to have some biopsies from patients with diabetes and skeletal muscle biopsies from patients with diabetes, and we looked at miR29 expression, and you can see that the levels of expression are higher in the diabetic patients. So next, using a high-fat diet model, or closer to a type 2 model, we wanted to see what happens with miR29. Again, in the ischemic non-diabetic, miR29 expression goes down to a lesser extent in the diabetic or high-fat diet-fed mice. We see impaired ADAM12 expression. And if we now ask the question, well, what happens if we inhibit the miR29? Can we improve ADAM12 expression? And the answer is yes. This is an experiment in which, this is the ischemic tissue in a control animal. This is the diabetic animal that has impaired ADAM12 expression. If we put in more ADAM12 using cDNA approach, you get some increase in ADAM12 expression. If we inhibit miR29, you actually get a very robust increase in ADAM12 expression, as you can see in the Westons. Then we looked at those animals that were treated similarly for what happens to perfusion recovery, and you can see inhibition of miR29 actually has a robust effect in restoring or improving perfusion recovery in the diabetic animals. And of course, one of the reasons why we were interested in this is because of potential use of microRNAs for therapy in the human disease. I want to point out that I recognize that we're only looking at one target of miR29, in this case, ADAM12. I want to emphasize that I don't think that all of the effects of miR29 is occurring through ADAM12. MicroRNAs tend to target multiple genes within a similar pathway. So the effect that we're seeing, yes, it's impacting ADAM12, but ADAM12 is not likely the only gene in that pathway that the microRNA is impacting. Which is also the reason why they can be more potent tools to be used in therapy. So next, let's see. So in the same, from these mice, we looked at the extent of capillary density, and you can see that, yes, you get increased capillary density in the mice that were treated with both ADAM12, both in the mice that were treated at ADAM12 and those mice that were treated with the miR29 inhibitor. You also see improved or muscle myopathy in those mice. This is the block 6, high-fat diet-fed or diabetic mice. You can see a lot of centralized nuclei, less so in the ADAM12 and the miR29 treated tissues. So just to summarize what I've told you so far, ADAM12 is a gene within LSQ1 and plays a key role in post-ischemic adaptation. ADAM12 is regulated by MIR29 through direct interaction of MIR29 with ADAM12 mRNA. High-fat diet or diabetes alters MIR29 expression, and inhibition of MIR29 in the high-fat diet or diabetic mice improve post-ischemic adaptation. I'm going to switch quickly and tell you about another story that we've followed a little bit, and that is that of BAG3. So what I'm showing you here is after our initial mapping of the limb salvage QTL1, these are the genes that we found within our refinement of the initial mapping. One of those genes is BAG3, shown here. And the reason why we were interested in BAG3 will be obvious in a minute. Again, for those who do this, you'll see that let me go back. Yes. You get a lot of genes, and then you have to decide how do you pick which one's of interest and which one do you pursue. And for us, one of the reasons why we picked this is because BAG3 is a 575 amino acid member of the BAG family of proteins. It is expressed in all mammalian tissue, but most prominent in the heart and skeletal muscles. Okay? And of course, as you know, the target organ in PAD is the lower extremity skeletal muscles. BAG3 deficient mice develop a fulminant myopathy in their muscle. Okay? In humans, defects in BAG3 have been linked to severe childhood muscular dystrophy. And one of our collaborators, Joe McClung, actually showed that a variant of BAG3 that's expressed in a BALB-C mouse. The point he tried to make in this study was basically that the BALB-C expresses, if you remember in the beginning, I showed you two strains. One that responds well, which was a Black 6, the one that doesn't respond very well, that has a tendency to lose its limb. What he showed is that that mouse expressed a variant of BAG3 that seems to be less effective in adaptation to injury, and therefore, when he put in the Black 6 variant of BAG3 into that mouse, he was able actually to improve the muscle myopathy in that mouse. He also went on to show that if you actually isolated skeletal muscle cells from those mice and cultured them and exposed them to ischemia, that the skeletal muscle cells from the BALB-C were more likely to die and exhibit muscle injury. So the first thing we did, trying to better understand the role of BAG3 in diabetes-related diabetic PAD, is do a time course of BAG3 expression in the ischemic limbs. And what you see here is there's actually less expression initially, and then there's upregulation over time with a peak expression around day 14 after the limb injury. However, if you look at in a model of diabetes using the high-fat diet and STZ model here, you see that there's less BAG3 expressed in the ischemic muscle from ischemic limb. This is whole gastroc being looked at here of those animals, right? And that's confirmed. You have similar impaired expression, whether you look at the mRNA at day 7 or day 14, and of course, we're looking at the protein at day 14. If you look at non-ischemic tissue in those mice, we don't see much difference in BAG3 expression. However, again, in a slightly different model where it's just high-fat diet feeding, you have the same issue. There's less BAG3 expression in the ischemic limb. Then we ask the question, we already know that these mice have a tendency to lose their limb after high-limb ischemia surgery. What happens if we restore BAG3 expression? So using an AAV-based approach, we were able to, under the control of a CNV promoter, we were able to restore BAG3 expression in those ischemic diabetic limbs, as shown here. And yes, we were able to rescue a lot of limb loss that these mice tend to execute, as you can see here. We also saw an improvement in perfusion recovery. When you look at the tissue sections from those mice, when stained for E-myosin heavy chain, which is a marker of regenerating myofibers, you see there's increased staining for that. When you look at capillary density, you see that there's increased capillary density in the mice that are treated with BAG3, and less skeletal muscle myopathy in the mice that were treated with BAG3. So the next question we ask is, what is the mechanism? What's the molecular mechanism of BAG3 action? When we look at the literature, there's been some evidence in the literature that BAG3 might impact autophagy. And so we looked at autophagy markers in the high-fat, ischemic high-fat diet tissue, and you can see that there's decreased expression of both ATG5, ATG7, and the LC2 to LC1 ratio is also very diminished. At the same time, in those same tissue, there's increased evidence of necroptosis markers, looking at RIP3, RIP1, and MLKL, suggesting that in the high-fat diet-fed mice, in their ischemic tissue, there's less autophagy, increased necroptosis. When we looked at the same parameters in mice that have been treated with BAG3, where we both expressed BAG3, you see now a robust expression of BAG3, first of all, and that was associated with increased ATG5, ATG7, and the LC2 to 1 ratio was increased, so consistent with increased autophagy. And this was also associated with decreased markers of necroptosis, looking at RIP3, RIP1, and MLKL. So the next question we ask is, does hyperglycemia and diabetes negatively impact ischemic human skeletal muscles? And so we took primary human skeletal muscles cells and grew them under high glucose or high glucose plus palmitic acid to try to simulate what happens in a diabetic milieu. These cells were then exposed to what we call simulated ischemia, where you drop the oxygen content and also reduce nutrition so that in order to simulate ischemia. And what you see is that there's actually increased death of those skeletal muscle cells under high glucose and palmitic acid. Under those same conditions, when you look at BAG3 expression in these cells, you can see that there's less BAG3 expression, as quantified here, suggesting that high glucose or high glucose plus palmitic acid impacts BAG3 expression in ischemia and contributes to increased death of the skeletal muscle cells. Here, what we did was actually took skeletal muscle cells from patients with diabetes now and went through the same thing. If you grow them in high glucose or high glucose plus palmitic acid and then expose them to ischemia, what you find is, again, increased cell death. When you look at BAG3 expression is also low, consistent with what we find in the non-diabetic tissue grown in high glucose as well. So how does diabetes or high-fat diet impair BAG3 expression in ischemia? That's one of the key questions we've been trying to answer, because we see impaired BAG3 expression. To do that, we first had to have an understanding of how does BAG3 expression gets upregulated in ischemia to begin with. And so what we've done here is a classic promoter bashing, where we're taking the promoter segments of the BAG3 promoter, constructed it, put in front of behind a reporter, in this case luciferase, and then we made smaller pieces of that promoter, and then you transfect this and look for report activity. And what you see is regions you see robust expression when you have robust luciferase activity when you have the full length. And as you lose different segments of the promoter, you can see loss of activity. This allows you to then identify regions of the promoter that are important. And then you can look at the sequence of the promoter and identify transcription factors that bind to those regions to try to begin to identify transcription factors that might be of interest to study in terms of BAG3 expression. And so one of those regions is right here. You can see a huge drop in activity here. And that's an area that has SP1, two SP1 sites and an EGR1 site. Interestingly, this area has a loss of EGR1 site and did not have a major drop in activity, suggesting that this is most likely due to the loss of SP1 sites here. So we asked the question, what happens to SP1 expression under these circumstances? Under ischemic condition, you see upregulation of SP1, just as you see upregulation of BAG3. And that's just a quantitation of the Western here. So we asked the question, what happens if you knock down SP1? What happens to BAG3 expression? So here we're using siRNA to knock down SP1. You can see that also results in decreased BAG3 expression. And that's the quantifications shown here. Next thing we did was go back to that same promoter construct and look for activity of the using the BAG3 promoter, driving luciferase. Look for that activity under ischemic conditions, and you can see that you have increased promoter activity. If we then take SP1 cDNA and transfect it into the cells that have been transfected with this reporter, you see increased expression or increased report activity. If you do that under ischemic conditions, you see an even more robust increased activity of the reporter. What this taken together suggests is that indeed SP1 is a transcription factor involved in driving BAG3 expression, especially, and this is more active under ischemia. So we asked the question then, in the same human skeletal muscle cells where we've shown increased SP1 activity, what happens if you grow those same cells with in high glucose and palmitic acid and you expose it to ischemia? You can see there's less SP1 activity. And you go into the mouse with high-fat diet and STZ, you take the ischemic tissue, we see again less SP1 protein being detected. To then try to understand why, we went in the literature, and one of the things that's been shown previously is that increased phosphorylation of PKC beta in endothelial cells plays a role in the pathology or the impairment of endothelial function in diabetes. So we asked the question, what's happened to PKC beta phosphorylation in these cells, in these muscle cells, skeletal muscle cells? And what you see here is that under high glucose or high glucose plus palmitic acid, you see increased PKC beta phosphorylation, and that's shown here. When you go in vivo, and no, this is still in vitro, both under normoxia, that's what this is, and under ischemia, you see both evidence of PKC beta, increased PKC beta phosphorylation. Then we asked the question, what if we inhibit PKC beta phosphorylation in these cells? What would happen to SP1 expression? And that's what we're showing here is, again, this is the model. High glucose phosphamidic acid leads to increased PKC beta phosphorylation. If you inhibit using riboctostirine, you see less PKC beta phosphorylation, increased SP1 expression. And this is just the quantification of the bands here. See less PKC with riboctostirine treatment, you can see less PKC beta phosphorylation and increased SP1 expression. Then in vivo, we asked the question, can we actually detect in the diabetic mouse tissue increased PKC beta phosphorylation? And the answer is yes. I think that's all I have. So in summary, the metabolic environment in diabetes alters mRNA expression, resulting in impaired post-ischemic adaptation in glial cells. Impaired upregulation of BAG3 in ischemic skeletal muscles is mediated by PKC. At least our data suggests it's mediated by, at least in part, by PKC-mediated mechanism. And these are the folks in my lab that's done the work. And when I give these kind of talks, I like to acknowledge those who trained me and those who've been involved in my mentoring over the years. So I trained, I'm an endocrinologist who went into a cardiologist's lab. Brian Annex was the cardiologist that trained me in vascular biology. And Dale Abel, Sam Dagogo-Jack, Kwame Osei, and James Garvin are endocrinologists that have, over the years, been involved in my mentoring. And the work's been supported by Foundation and the NIH. Thank you. I'll take your questions.
So those online, you can put them in the chat box, but otherwise it's open.
So you showed early on in the diabetes-induced disease that you could prevent disease with insulin or insulin therapy, and you didn't talk about how well controlled, right? But have you ever gone back in any of these later models and used insulin to restore normal?
So,
and what does insulin do to those targets that you identified as well?
Yeah, I wouldn't say we can prevent disease. I would say we restored VEGF receptor. We prevented ubiquitination of VEGF receptor in that context. We didn't look at more broad physiology. And so in that experiment, what we're trying to show, or at least what we're trying to test, is in vivo, if you control for hyperglycemia, can you control for the increased ubiquitination that you see in that setting? So that was the extent of those studies.
So the reason why I asked the question
Sure.
is it gets back to the human tissue you use to correlate the animal findings with the human findings. And if the individuals that provided the biopsies, which is probably not the case, were well controlled, I would be surprised. It would be surprising to see those responses you saw. So I'm guessing that those individuals, the human tissue, the biopsies, were probably from uncontrolled diabetics?
Yes, that is correct. But you're asking a very important question, which is, can control of diabetes prevent this?
Right.
That's a very good question. And the answer is, I don't know. Because I have actually not seen any data that carefully looked at PAD in diabetics with excellent glycemic control.
Maybe it didn't happen.
That's possible too, right? So what we've seen though is data that looked at among people that are poorly controlled. You have more severe disease if you have higher A1C. So it's still in the milieu of poor control. The worse the control, the worse they do. But go ahead.
That also leads to the question of obesity versus diabetes, right? And is there an association with disease in obese patients independent of the diabetes?
The answer is yes. Obesity, insulin, there's at least one study that I'm remembering right now that showed that just the presence of significant insulin resistance was associated with PAD. And those patients were not diabetic.
So that's really interesting, right? So that suggests that it's insulin's really important in this model.
Yes.
And not just the excess fatty acid associated with obesity. And it's really driven by the absence, potentially, of insulin signaling. So
but hyperglycemia by itself is able to, at least in vitro models, it's sufficient to drive it. But I hear you. I had the same questions churning in my head when.
We're kind of saying the same thing in a circle, right? Okay. This is one of the challenges, but it's really cool to think about. I mean, do PPAR gamma ligands, have you ever looked at the effects of PPAR gamma ligands in this model to see if you can actually
Restore.
Yeah.
So earlier in my career in the vascular lab, I actually got pioglitazone and was feeding it to the mice, and they didn't do too well. So I was never ever able to follow through. Yeah. I'm trying to remember why now. I mean, of course, there was tablets that we just simply obtained. It wasn't like formulation for mice or whatever. It's basically a prescription for pioglitazone from humans that we were grounding up and were feeding to mice at that time. I never followed it further because the mice were just not doing so well, and it was hard to, you also have to do daily gavages and,
Yes.
yeah.
Metformin may be another one that would be worth going after, because a lot of your gene hits were associated with metabolism.
Yes.
Metabolism.
Yes, I think, that to your question, one of the key questions that I've struggled with is this idea that even in a type 1 model, because we know even in the type 2 models, it's a little more complex, but even in the type 1 model, is it an insulin deficiency or is it hypoglycemia?
Right, this is the right.
And it's really difficult to separate the two. And I actually started doing some experiments where I was trying to use SGLT2 inhibitors to try to see if I can drop blood sugar without providing any insulin. And again, back then, it was an injectable form that was available, and the mice would develop these really bad skin stuff that the mouse facility folks were not allowing me to keep those mice around because they get bad skin disease that you have to come back to mice. Right? So we never were able to follow them through. These might be experiments that we can revisit now that we have better SGLT2s that are available in humans, and we might be able to introduce these mice.
So one last question, and I'm sorry I'm hogging the entire time as usual.
That's why I looked at you first.
So one of the things, we use strepsitosin all the time, right? Everybody uses it to induce diabetes. No one wants to talk about anything other than strep kills beta cells. But that's really not true. It causes liver damage and it causes vascular damage. And are you driving disease because of the vascular damage of the strep versus does that initiate disease? And so you did the right experiment. And so I'm interested, does the Akita mouse look just like the strep-treated animal? Because the Akita mouse gets around that issue.
Yeah, good question. I never did side by side, and even with the high-fat diet model, I started with just high-fat diet for that very reason. Reviewers criticized my grant, said you have to use STZ, and since I'm not going to be in the room to argue with them, I simply use STZ plus high-fat diet. I mean, but I agree with you. STZ's effect is not just on beta cells. The reason why it kills beta cells is because beta cells concentrated more and they're more likely to be injured by it, but it can knock off other cell types. But I mean, even within my division, when I bring that up, it's like, someone's like, shut up. We all use STZ, so.
Mess it up.
Exactly. It's like, no, it remains a concern.
So so you have two pretty interesting proteins that do very different things in terms of ADO12 and BAG3.
Yes.
And you're doing this in a really complicated system because you have muscle, you probably have some fat in those tissues, you have endothelium, you have the immune system, you have a history of immunology. So where is ADO12 expressed and what's the sites that it's working at? And
Beautiful question.
how do you tease that out? Or you can do the same thing with BAG3, and I mean, I have ideas about BAG3, but.
So I think those are outstanding questions, right? And the answer is ADAM12, our initial studies focused on endothelial cells because when we take the muscle and we actually separate out endothelial cell fraction, we just concentrate for it because you can't get rid of. We enriched for endothelial cells. We saw that in ischemic muscle, a lot of the upregulation is occurring in endothelial fraction, and that became why we went down the endothelial route. However, skeletal muscle fiber expresses ADAM12, so I can't rule that out. It is not until now actually, I must say that I actually have mice to test this, where I am able to knock down atom 12 specifically in muscle, endothelial cell, vascular smooth muscle cells, so that we can now revisit this question of where is it important in this milieu. On the BAG3 side, because what we see is low expression and we're doing gain of function studies, we made a TET responsive BAG3 mouse that we can give doxy and will express a BAG3 specifically in the muscle fibers. And so hopefully we'll be able to now be able to explain, is it muscle, is it, you know.
Just be a little bit careful with docs because it'll also inhibit PKC, so.
That's good to keep in mind. So we may have to.
No one
Yeah.
thinks about it, right, but it's been well known for a long time. Oh, actually, it's tamoxifen that does that, but it's the same kind of thing.
Right.
All right, I won't ask her anymore.
You have a question? So I was wondering, it's a chronic disease when you miR-29 therapy. What was the duration? How did you give it? How does that ischemia? Because it's not very like how many days and.
Yeah, so because we're using AAV, you will get expression 4 weeks, which in mice, that's a long time because without any intervention, when we cut out the femoral artery, the mice will be fully recovered in 3 to 4 weeks for the strains that recover very well. AAV-mediated overexpression lasts several weeks. I mean, people have shown as much as 2 months, right? So we know we get long-term expression. Related to your question, though, is if we're going to use this as therapy, right? How do we get it into the muscle? Are we going to use AAV? Probably not. One of the approaches we've been trying to use is this microbubble mediated delivery, where you inject systemically, you burst in a tissue of interest, and you get. So you can modify the microbubble a variety of different ways to target endothelial cells and so forth to get it into the tissue. And we've had some success with that. The problem, again, is in mouse, the whole gastric is not that big, right? So you can hit the whole gastric all at once with an ultrasound probe, and I can get. In humans, it's still better than doing 20 or 40 injections of AAV into the tissue. If something like that can work, I can see where you can do a systemic infusion of something, and then you burst it in the tissue of interest because, fortunately for us, muscle is right there, and you might be able to actually get robust expression. One of my next steps is actually to do large animal models first before we start talking about humans. So I have a collaborator that we're trying to do some of these things in pigs and seeing whether proof of concept we actually can get systemic delivery, robust expression locally. But it looks like you have another question.
No. It makes
No,
perfect.
yeah, no, I mean, it's
in Iowa.
it's an acute limb ischemia, but in humans that it is a chronic, it's atherosclerosis or whatever diabetes, it's stopped decades. So would you expect similar recovery?
This is one of the key questions in the PAD field, though, that the average PAD patient who presents with claudication has chronic and partial occlusion of their vessel, right? But the patient who presents with peripheral limb ischemia, I think, is a little different. Often they wake up and it's blue toe, or they see pain suddenly occurs, and different people, of course, handle that differently. Some, like the guy I just saw last week, called the PCP, called the doctors, blah, blah, blah. After a day or so of the toes increasingly getting dusky, and they said, come in the hospital, and they started trying to do something. Some people will just sit with it and hope it goes away until the toes are black, and then they seek medical help. What I'm trying to get at is that the patients who have limb-threatening ischemia, I think, often do get acute limb occlusions. it could be that there's some chronic process that has been ongoing, and then you hit a critical point. We don't know what happens there is what happens in the heart, where you have plaque rupture that actually closes up the vessel, and of course in the heart you get pain. So you know to go get some help because chest pain occurred, so you seek help. In the limbs, does plaque rupture occur? Nobody's really demonstrated it. It's assumed that atherosclerosis in the limb is similar to in the coronary vessels, but that might not be the case. There's some data that suggests the endothelial lining of the coronary vessels is very different from the endothelial lining in the lower limbs. Do we expect them to behave the same way? Maybe not. The pressures in the heart, filling the heart vessels, is very different from pressures in the leg. And at least when it relates to collateral formation, it's clear that pressure matters in terms of the behavior of the endothelial lining. So yeah, there's a lot of questions that remain unanswered. But as far as the model relates to the disease, there are the different form of the model that's been developed where you deploy amyloid constrictors that will squeeze, and get you occlusion over a longer period of time. I think in terms of the diabetes, no matter what method you use, the diabetic animal does worse than the non-diabetic. So in terms of pathology, it's similar.
So where do you think you have? So in this disease, where do you think you need to get if you're going to use the siRNA or the microRNAs, where do you need to get it? Because AAV may be perfect because you can track where the occlusion is. And if you need it at the site of the occlusion, right, then you should be able to do the injection right there.
I think in a non-diabetic, you have tight occlusion.
Sure.
You can tack that. In the diabetic, it's microvessel, it's very diffuse. So, but you're right. We still need to. What I like about the AAV actually is the fact that I can tag with a dye. I can actually go back and look. To what extent are we getting expression of this AAV? Because not only a gene of interest, because we often will put a reporter like a GFP or HA tag or something at the end of that, so you can actually see where and to what extent did you get the tissue, expression in your tissue, not just by taking the whole thing. You can do a section and you can see how much of it. So yeah, but I think to get a sense of how that might work in a human large animal model is, you can get the hind limb of a pig, 80% of it or 50% of it to be infected and get expression. You might have a very different pathology than if you don't.
Thank you.
Thank you.